Hi there guys, and welcome to another A Level Biology video with Mr Phillips. This is video year 12 1.11 now, which is going to be on DNA replication, which occurs in all living organisms. So continuing down the nucleic acid part of biomolecules, and we're just going to focus on DNA replication. So just to remind you, DNA is a stable information storage molecule, which is obviously extremely long, a lot of information in there. It's made strong because it's got purines and pyrimidine bases, and the complementary base pairs of A to T, G to C, with 3 hydrogen bonds between G and C and 2 hydrogen bonds between A and T. And obviously that complementary base pair rule is going to be essential in copying the DNA and making more DNA for our cells when they replicate. And that's just a bit more on that again. So why does it need to happen? Well, it happens during interphase. So if you haven't done the cell cycle yet, you will have no doubt at GCSE talked about, you should have talked about cell cycle at GCSE, but you have talked about mitosis, which is when you make 2 genetically identical daughter cells from the original cell, and they have a full set of DNA. So this happens during interphase, and obviously each new daughter cell will need that full copy of DNA so it can become the same cell in mitosis, which we use for growth, to replace cells, and also in asexual reproduction of some organisms. And this allows the genetic continuity between generations of cells, and obviously eventually that will lead to new generations through meiosis. So let's have a look at that. So this is what I'm going to try and use to sort of talk you through this as we go through. So the first thing is you have a strand of DNA, and the DNA strand is millions and millions of base pairs in length. So one end of the strand can still be stuck together as a double helix, whereas the other end of the strand can be separated and being replicated. So it sort of unzips it. So it's a bit like if I've got my zip on, I'd take my zip down, one end of my coat can be open, whereas the bottom end can still be zipped up. Now, if we have a look at the stages, I'll try and talk you through them, and then we'll try and summarise them again at the end. But if you've got a question saying how does DNA replicate, if you wrote the information in this question and you understood it and explained it in sentences, you would get most of the marks, if not all the marks. So the first thing that we need to know then is with DNA replication is that a DNA strand, obviously it needs to be perfectly copied with whatever mutations, and that would be a big problem, and it happens during interphase. First thing though is the DNA is a double helix, so you can't really very well replicate something that's wrapped up in a helix shape. So first thing that needs to happen is the DNA untwists, and that is done by the enzyme DNA gyrase. And you don't need to explain how that happens, but that enzyme effectively untwists the DNA to make it much more like this ladder that we've talked about in the last video. Next thing that needs to happen then is if we're going to make new copies of DNA, is that we need to have exposed base pairs. So the strand basically has to become unzipped like that, so we can use one side to make a new DNA strand and the other side to make a new DNA strand. So you end up with going from 1 to 2 DNA strands, and you go from double to single stranded in that part of the DNA strand. And in order to do that, we need to unzip the double helix, so we're breaking the hydrogen bonds effectively, and that is done using the enzyme DNA helicase. So again, another enzyme you need to know the name of. You've got 3, then in the nucleus there are 3 nucleotides. So what I mean by 3 nucleotide, if you look at the diagram, we go up to the top up here, you can see here there is a nucleotide that is not attached, so they're being made constantly in the nucleolus, I believe it is, and they go into the nucleoplasm, and then the free nucleotide is then able to attach. So you can see, if you see with where this diagram's going, sort of the hydrogen bonds are still unzipping below this purple line, but above the purple line they have unzipped. So then we've got a C here, and that C is obviously going to attach to that G there because it's complementary, and as you should be aware, they'll form 3 hydrogen bonds between those 2, and then we'll need like an A to go there, and would need a nucleotide with a T to go there. So whilst it's obviously complementary base pairs, it is a free nucleotide because there's a phosphate, a sugar, and a nucleic an organic base. So this is the point where, as I said in the last test, you always need to mention the idea of the complementary base pairing. So say A to T, adenine to thymine, G to C, because it may well be worth an extra mark for you. So once you've then got, if we have a look at this right hand chain here, so this is like one of our new strands here of DNA. And what we want then is we need this strand, the new strand coming in, the lagging strand coming in. We want this one to basically combine. So you see you've got the phosphodiester bonds combining the phosphates, the sugars here, but then we're missing these last two here. So the next thing that has to happen is we have to have a condensation reaction, which, if you remember what that is, it is the removal of a water molecule, which is one of the vital reactions that control all biological molecules combining. And the condensation reaction then combines the phosphate of one molecule, of one nucleotide, to the sugar of another nucleotide. And that is then your phosphodiester bonds being made, which make the new backbone of the DNA structure. Now that is controlled by the molecule DNA, the enzyme, DNA polymerase. And obviously, as the new bases are as that is forming, you can keep adding new ones onto the end. So then we've got the, to have a look just a bit closer at DNA polymerase, the enzyme that controls the building of the new DNA strand. So the hydrogen bonds have already formed at this point between the complementary base pairs of the new strand and the template strand which we're using. So here you've got the G, and that's obviously combined with C, and they'll have 3 hydrogen bonds. And then we've got the A combined into the T, which will have 2 hydrogen bonds, and so on and so on. So that's all sorted. You don't need to talk about that. You could mention in the exam question that the purines and pyrimidines, that would obviously be acceptable as well. Now, if you look at this picture here, you might not be able to see as clearly as I'd like, but there is a molecule here of DNA polymerase which is covering both strands. Now, the DNA polymerase is an enzyme, okay, and it's actually working on attaching. It isn't actually working on the old strand. So if you think about it, the DNA polymerase doesn't actually do anything to the old strand because the old strand's already built. So it hasn't got to combine phosphates and sugars in the old strand for a condensation reaction. It has to work along the new strand. So the active site of the DNA polymerase is effectively, if you think about it, sort of like here. Okay, and that is because the new strand has to be built from what we call the 3' end of the DNA strand. So DNA strand, based on where the sugars are facing, have a 3' end, which is where they've got an OH group, and a 5' end, which has a phosphate. And the active site of the DNA polymerase can only work on the 3' end of the new strand, where it attaches the new free nucleotide to each bit, making the new strand longer. So the DNA strand will then only grow in the direction of the 3'. So the DNA polymerase is moving down towards the 3' end of the new strand. And obviously, as I said before, it's controlling the reaction, it's doing condensation reactions, adding phosphodiester bonds or ester bonds to between the nucleotides. Now, what that then leads to is the type of replication. So basically, when they worked out what the structure of DNA was, a lot of questions were, well, how can it actually replicate? And there's 3 potential models that they came up with. So we're going to discount 2 of them straight away, and then we'll look at the evidence for the actual one they've accepted. So the first model, if we look at C, the dispersive model. So the dispersive model is this idea that if you take a piece of parental DNA, so the original piece of DNA, and obviously new DNA has to attach to that to make double the amount when you replicate DNA. And what they said is that it would be dispersive. So here you see this bit, if you look on the left hand side, you've got a bit of purple DNA, followed by a bit of blue DNA, followed by a bit of purple DNA, followed by a bit of blue DNA. And the idea was it might interlink. That one seems very odd that would have even possibly happened. So that one was discounted very quickly. Then we've got the conservative model. So the word to conserve means like to conserve animals. We conserve all the animals, would save all the animals. So the idea of conserve is that the original strand of DNA doesn't change, and you end up with one original strand of DNA to one new strand of DNA. And then if that went on into a generation, your original DNA would fade out to a certain pattern. So you would then have from that one, two new strands of DNA, which are completely new with none of the original parts. And then this one, you'd have one new, one old one. But that has also been misjudged. And then you've got semi-conservative replication, which is the idea here that if you've got one old strand, you've got the original strand of DNA, the parent on purple, then one of the strands on each of the new DNA, one of the two anti-parallel strands is the original DNA, one of the new strands is the old DNA, and that is a model that's accepted. But how do we know that? Well, you would use something called the centrifuge to understand this. A centrifuge is a electronic piece of equipment which spins at high speed, and it allows us to separate molecules and allows us to separate biological molecules such as cells and DNA according to their mass. So the experiments that we're going to look at prove that semi-conservative replication happens was from Measles and Stahl in 1957. And what they did is they grew E. coli on heavy nitrogen containing agar and heavy nitrogen, so it's an isotope of nitrogen 15. So normally nitrogen has a mass number of 40. With an extra neutron, it becomes M15. And what they did is they used a centrifuge to then spin and work out the weight of the DNA when they added it then to nitrogen 14. Now what you would expect is this: if all the DNA if both strands of DNA were light, you'd have a red line as we're going to represent it up here, but your strand of DNA would fall there because of the mass. If both of the strands of the DNA molecule were heavy, you would get a heavy line at the bottom. If you had one light strand and one heavy strand, which would prove conservative replication has happened, you would get a line of light light and a line of heavy heavy there. But if you had semi-conservative replication, which means you've got one strand from the old DNA, the red one strand from the new DNA in your DNA double helix, you would have a light and heavy strand there in the middle. So what happened first of all was all of the molecules of DNA, or both strands were heavy, so you get this heavy line towards the bottom. Now obviously if we then add a lighter strand DNA, the next one to the next generation, if it was conservative replication, if it was going to make one totally new strand, you would expect to see this box here, okay, because that box there would obviously mean that you've got one original and then one new one. If, however, the new light nitrogen DNA nucleotides are combined with the heavy nitrogen DNA nucleotides for semi-conservative replication, you would expect to see that strand there, which is a light, heavy strand of the DNA together. So what did we see? Well, what we saw, so what did happen then? Well, let's have a look at it. So we took the original strand, okay, and if I use the colours we've been using on the other slide, so there's our original DNA strand, okay. Now our original DNA strand would have split into two because obviously it's DNA replication we just talked about. So there's your one original strand, there's your other original strand. My double helixes aren't great. They would then have attached to the new lighter nitrogen strand. So you would have got one piece of DNA with a single light and single heavy, and another piece of DNA with single light and single heavy. So what you'd expect to see is the next centrifuge result would have been that mixture, that purple line, that mixture of the two, and it was. So after two DNA replications, after first DNA replication, we got this result down here. So then what would happen next? Well, if you took those two, and those two would then divide into 4. So what we would do, if I draw a line here, is those two pieces of DNA would split to make 4 pieces of DNA. So you have the original 2 strands, which I can't draw very well, but also now you're going to be making 2 extra strands from the 2 new bits of DNA. And don't forget the new DNA is also red, it's also N14, sorry. So these two would have N14 attached to them to make a light heavy strand, but these ones which are N14 would now have new N14s, so you'd get a double N14 strand. So you would get a slightly different result for your next generation centrifuge. So if we have a look at that, you can see here you would get this result. So what does this mean? Well, the purple result here is these two, okay, where you've still got a strand that has got the original N15 that's being semi-conserved, but you've also, they're always attached now to a new strand, and then you've got this red line which is two new lots of DNA just made out of new strands of N14 which are here. And basically that original blue N15 never goes anywhere, but it's going to get cancelled out each time. So when these replicate, then it's going to be, instead of 2 out of 4 molecules have some N15, it's going to be 2 out of 8 molecules. And therefore what's going to happen is your thickness line on the N14 DNA, the light DNA, is going to get more, and your N15-14 line is going to stay the same. So effectively you'll see this starting to happen, and that red line will just keep getting thickness. Okay, so that's it for DNA. So we'll be looking at enzymes next. So I hope you found that useful, and I'll see you next time.